Okay, well, welcome everyone. Good morning. It's a real pleasure today to host Dr. Elvisha Dhamala. Who's going to give a special seminar. Elvisha applied for the AI scientist position at the KCNI. Dr. Dhamala graduated in 2021 from a PhD in neuroscience at Cornell University. And then she went on to do a postdoc at the Department of Psychology at Yale University until 2023, where she became an assistant professor at Northwell Health in the Department of Psychiatry and the Institute of Behavioral Science. Today, Elvisha is going to tell us about leveraging brain-based predictive models for precision psychiatry. So thank you very much for coming here, and we look forward to it.
Great. Thank you so much for having me. I'm Elvisha Dhamala, and I'm an assistant professor at the Feinstein Institutes for Medical Research in New York. And a lot of my research is about how we can leverage machine learning approaches and specifically brain-based predictive models for psychiatry research. So in Canada, at any given time, 1 in 5 youth are experiencing a mental illness, the most common ones being depression, anxiety, and behavioral disorders. Suicide is actually the 3rd leading cause of death in 10 to 24-year-olds, and 80% of those who die by suicide have an underlying mental illness. A lot of these illnesses tend to emerge at different points in childhood and adolescence, and prevalence actually changes with age. So here you'll see that something like anxiety is much more prevalent at the adolescent older ages, whereas behavioral disorders start off pretty high and then kind of remain more stable than anxiety or depression. If we look at lifetime prevalence of any mental disorder among adolescents, and this is data from the US, about half have experienced a mental illness at some point in their life, and about 1 in 5 have experienced a serious mental illness, meaning it substantially interfered with 1 or more of their life activities. Importantly, there's a sex bias such that different illnesses are more likely to affect men or women. So here on the left are the ones that are female biased or more common in females. Right is male biased, so things like attention disorders, autism spectrum, more common in males. Eating disorders, anxiety, depressive disorders, more common in females. And it's not just sex, it's also gender that has an influence. So specifically, if we look at gender incongruent youth, or youth whose genders do not align with their assigned sex, they have worse mental health than their gender congruent counterparts, and this is true for both sexes. So on the left, we have females, right is males, pink is gender congruent females, purple is gender incongruent. For males, again, the darker color is gender congruent, and then the other one is gender incongruent. And the dark black sections of the pie represent those that have these symptoms at clinically significant levels. So overall, you'll see that those prevalences are higher in the gender incongruent group than they are in the gender congruent group. So that's another factor that we have to consider that is above and beyond sex itself. And not only are these illnesses common in children and adolescents, that's also when they tend to appear for most people who go on to experience them. 50% of all of them emerge by age 14 and 75% emerge by age 24. And adolescence is a pretty broad term, so we also have to look at specific ages because there are different risks at those ages. So if we look at the distribution of age onset of mental illness in the general population, anxiety diagnoses peak earlier at ages 5 to 6, and then they also have a second peak at around ages 15 to 16. OCD diagnoses peak around 14 to 15, eating disorders a bit later, and then things like substance, personality, and mood disorders come after that. Adolescence is also a very important time for brain development because the brain continues to mature throughout this time in terms of gray matter, white matter, connections, and everything like that. And it's not just that the brain is changing at a structural level, different neural and cognitive systems that are relevant for mental health are also going through specific sensitive periods where they're highly plastic. So sensitive periods are typically time-limited developmental windows where certain aspects of internal and external influences on a given system are more likely to have an effect than at other times. And unsurprisingly, those sensitive periods also line up with when different mental illnesses emerge. So you'll see that the threat system has a sensitive period that's earlier on. That more lines up with something like anxiety, Something like prefrontal cortex and overall functional connectivity is a bit later. That lines up with more mood and personality disorders. And then social cognition, which comes later, lines up with things like personality disorders and schizophrenia. Obviously, it's not perfect, but it does suggest that depending on when we experience specific types of adversities and how those affect these different systems, that might also have an effect on the types of illnesses that we go on to experience. So all of that to say that these illnesses are highly common in youth, and that's when they tend to emerge for a lot of people. But even though many of these illnesses tend to begin to emerge during this early stage of life, the average delay between onset of symptoms and treatment is actually 11 years. So symptoms might start pretty early, diagnosis is much later, and then treatment itself can take even more time. So instead of thinking about it as an overall, we have to actually think about them as specific stages. So the premorbid stages are when there are often no or few symptoms. And in the prodromal, we see some symptoms beginning to emerge. Syndromal, we see them at a clinically significant level. And then chronic stages are typically when some sort of plateau is reached. But adolescence is not just a time when things can go wrong, it's also when a lot of things can go right. So adolescence is a time when individuals can develop mental well-being, and this is through things like good health and optimum nutrition, safety and a supportive environment, having learning, competence, education, and overall things like that. But you can have mental well-being and mental illness at the same time. And specifically, a lot of the structural changes that are happening in our brains, which also underlie the functional changes that are happening, are mediated by biological and environmental factors that stress can then affect. So different aspects of myelination, interneuron maturation, connectivity, dopaminergic projections, all of these are still developing, and depending on when in the critical period we have specific stressors, that will influence when these illnesses actually emerge. Importantly, there's sex differences in this structural development pattern, such that males have larger overall brains and females tend to reach developmental peaks 1 to 2 years earlier, but this maturation is not uniform. So throughout childhood and adolescence, cortical development progresses from a lower order primary sensory cortices to higher order transmodal association cortices. And we see this for cortical thinning, intracortical myelination, white matter development, functional refinement, and so on. So this tells us that there's also a gradient. So depending on when the actual exposure is happening, different parts of the brain are more likely to be affected in terms of sensory versus association. At the same time, these association networks also tend to differ across the sexes. So these are the feature weights that you get when you classify individuals for sex based on their functional network topography. And you'll see that the highest weights are assigned to things like the ventral attention, default mode, frontoparietal, and dorsal attention. So this tells us that those same networks that are developing later on are associated with mental illness are also different across the sexes. And specifically in terms of mental illness, neural disruptions in these cortices have been implicated in a lot of different mental health traits and illnesses. So at the bottom, we have alterations in cortical thickness, and it's very strongly related for bipolar disorder and schizophrenia. On the top right, those are the feature weights you get when you use functional connectivity to predict mental health traits. So again, you'll see that there are a lot of high weights in those association networks. And then finally, hypo and hyperconnectivity has been linked to a wide range of psychiatric diagnoses in a very transdiagnostic manner. So given that a lot of these aspects are changing throughout adolescence and there are sex differences, I'm now going to talk about the brain and how we can actually leverage these brain-based predictive modeling approaches to understand adolescent mental health. White matter fibers in the brain directly and indirectly connect different brain regions, and these structural connections facilitate the flow of neural activation signals between brain regions. These structural and functional properties of the brain underlie our unique and individual behaviors. So it makes sense that if we look at variations in brain structure, function, and connectivity, they're associated with individual differences in psychiatric diagnoses, dimensional symptom profiles, cognitive abilities, and personality traits. Traditionally, these relationships have been studied at a group level to identify differences between populations like healthy and patient groups, males and females, children and adults, and so on. And that's helped us gain a lot of understanding about the structural and functional underpinnings of illnesses and behaviors. But with increased access to computational resources and large publicly available data sets, we can implement data-driven machine learning predictive models to better understand these relationships at an individual level. In doing so, we can predict things like illness risk, disease progression, treatment response, and so on better for each individual. And what's great is that not only can we make these individual predictions, we can also leverage these approaches to gain a mechanistic understanding of the neural bases of different illnesses. And these models, as their name suggests, relies on neuroimaging. So neuroimaging is used to directly or indirectly image the structure or function of the brain. I know a lot of you are familiar with this, so I'll go through this very quickly. Varies in temporal and spatial resolution. I care about whole brain, so I primarily use MRI data. This includes structural MRI, which qualitatively and quantitatively describes the shape, size, and integrity of gray and white matter structures. And through this, we extract things like surface area, gray matter volume, cortical thickness, which we then link to different behaviors. Diffusion MRI maps white matter pathways in the brain, and using the diffusion data, we can calculate what's called structural connectivity. And this represents sorry, I thought I was sharing a video. Structural connectivity represents how connected two brain regions are in terms of actual white matter fibers, so actual anatomical connections. And finally, we have functional MRI, which indirectly measures brain activity by looking at blood flow patterns. And we can use those to look at functional connectivity, which represents a statistical correlation between regional activation patterns. So structural is anatomical, functional is statistical. And we can use these structural, functional, and diffusion data in combination with machine learning algorithms to develop these brain-based predictive models. So machine learning as a whole is the use and development of algorithms and models that are able to learn and adapt without following explicit instructions. I'm not going to go through the whole process of how that's done, but I will talk about 3 different types, and those are descriptive, predictive, and prescriptive models. So descriptive analyses, or descriptive models, identify underlying patterns in the data to describe and summarize it in an easily interpretable manner. So they do so by looking at how data might cluster together, and it can reveal broad trends in neurobiological, behavioral, or clinical phenotypes. And these are unsupervised, meaning that they're not trained on labeled data. So this approach has been used to identify distinct subtypes or biotypes of patients with depression based on functional connectivity. These clusters are shown in red, blue, purple, and black. And not only do these clusters kind of vary in terms of their functional connectivity patterns, they actually also reveal distinct sets of symptoms. So if we look at anhedonia, anxiety, insomnia, and fatigue, they're different across these different clusters. So even though all of these individuals have the same depression diagnosis, their functional connectivity patterns can be used to distinguish clusters that differ in their symptoms. Next, we have predictive analyses, which establish associations between independent and dependent dependent variables, and use those associations to predict trends and properties in unobserved samples. So these are supervised models, meaning that they are relying on pre-observed ground truth labels to then make individual predictions. So this approach has been used, for example, to predict borderline personality disorder in children and adolescents. So basically looking at functional connectivity, they were able to train it to predict these BPD scores, and then they were actually also able to look at the specific networks that contribute to those behaviors, which is shown in the bottom right. Finally, we have prescriptive analyses, which allow us to recognize risks and offer potential treatment solutions at an individual level. So these identify specific anomalies in the brain using the brain imaging data, and then can actually suggest personalized interventions that can most effectively alleviate those specific concerns. So one example of this is a biomarker that's been identified that's called the SCI, or the Striatal Connectivity Index, which actually predicts response to antipsychotic drug treatment in first episode psychosis. And collectively, these descriptive, predictive, and prescriptive analyses can be used to identify disease-relevant subtypes, predict symptom profiles, and recommend personalized therapeutics for individuals. So overall, these capture multivariate associations And these multivariate associations can be used to advance mechanistic understanding and make individual predictions about mental health outcomes. I'm just going to take some water, sorry. But when we develop these models, it's important to recognize that the methodological choices we make can influence how accurately they can capture brain behavior relationships and the extent to which we can actually interpret those relationships. These choices include things like the neuroimaging modality and the state that we choose to base our predictions on, the parcellations we use to segment the brain, data transformation approaches we use to apply to the data before it gets fed into the model, the sample size, whether we're using population-specific or general models, the algorithms that we're using, and specifically the phenotypes that we're trying to predict. So a lot of the work that I've done has addressed different aspects of these methodological choices in combined or separate ways. So I'm going to go through different aspects of the methodological work that I've done, and then eventually I will get to specific studies where we've used these methods to actually predict clinically relevant behaviors. So in this first study that I'll talk about, we looked at something very simple. Does structural or functional connectivity better predict cognition? And this was specifically in the HCP sample. And we were looking at crystallized and fluid cognition. And here I'm showing the explained variance, and the colored in violin plots indicate that they perform better than chance. We also used what we called a hybrid connectivity, which just concatenated the structural and the functional data together. And what we found is that the functional connectivity data was always able to predict cognition. The structural connectivity was not necessarily able to. Importantly, when we combined the two into a hybrid connectivity, it actually didn't help, and it made the predictions typically worse than we got with just functional connectivity itself. As part of the analyses, we also looked at how you derive the actual connectivity has an impact, and we found that structural connectivity derived using probabilistic tractography is better than that using deterministic tractography. And I won't show another figure, but we also found that this sounds very obvious, but it took us a while to figure it out. Structural predictions are better when they're based on structurally defined parcellations, and functional predictions are better when they're based on functionally defined parcellations. So we also want to be careful and not just apply our same, whatever your favorite parcellation is, just broadly to all data types. So then, building on that, we wanted to see if the results from that previous study would also generalize across other domains of human behavior, as well as data sets. So here we were looking at sort of the same approach, but looking at cognition, dissatisfaction, and emotion in HCP, as well as cognition, personality, and mental health in ABCD. And overall, what we found is that functional connectivity does yield the most accurate predictions across all of these domains in both data sets. We also examined whether, again, results would be influenced by the specific measures within these different domains. So overall, we looked at different measures like thickness, area, and volume from anatomical data. Same for functional, we looked at rest and task data. And there's a lot of variation in how accurately they're able to predict behaviors, but there's not necessarily a clear winner that emerges. Similar results for the diffusion data and for the structural connectivity data. You might have noticed that for all of these, the cognition predictions are much higher than the rest of them. And that's just because cognition is much easier to predict than everything else. If mental health was easy to predict, we wouldn't be here. So a lot of what I'll show you in the next bit is focused on cognition, just because it provides us with a framework that we know will capture meaningful relationships that we can then see how these methods actually influence it. So for the next study, we looked at how correcting for ICV using a proportional correction can actually influence the accuracy and interpretability of those relationships. This was based on HCP and ABCD data. We did a lot of sex-specific analyses as well, given sex differences in ICV. And I'm not going to go into detail for the results, but we did find one really cool thing. So these are the feature weights that we get from the models to predict all of the different types of cognition, which is listed along the vertical, and then along the bottom, we have the different networks. And if we look at surface area and gray matter volume, and this is for HCP, surface area and visual networks and volume in default mode, language, and control networks were associated. And these results were pretty consistent with and without ICV correction. If we look at the same thing in kids, we see default mode and somatomotor for surface area and visual, somatomotor, and dorsal attention for volume. Again, pretty similar with and without ICV correction. Where it gets really interesting is with cortical thickness. So we actually see completely opposing gradients for kids and adults, and this gradient is only there with ICV correction. If you don't do it, it looks like some combination of these other ones. So this tells us that overall, for the actual results, in adults, heteromodal association networks were more important for cognition, but in kids, visual and somatomotor are more important. This also kind of reflects that sensory motor to association gradient that I showed earlier. So it suggests that cognition might be more dependent on areas that develop earlier on in life. And as other areas develop, the cognitive underpinnings might actually shift to those areas. And let's talk about algorithms. So there are classic machine learning algorithms, which are relatively simple models that can be linear or nonlinear and sparse or non-sparse in nature. And we also have deep learning algorithms, which rely on neural networks, either one or many, which progressively extract higher level features from the data. And although deep learning models can theoretically capture more complex relationships in the underlying data, simpler models are more interpretable. So as models become more complex, bias decreases, but variance increases. And there's also a trade-off between accuracy and interpretability. So a lot of times the models that theoretically can be more accurate will be very low in terms of interpretability. Those that might struggle with accuracy have higher interpretability. And while complex models typically can capture more specific relationships, there's a reason to believe that we actually don't need them, because in this study, it was actually shown that if you use a simple kernel regression, it performs just as well as several different types of neural networks. So even though we like to think we're very complex as individuals, sometimes a linear model is all you really need. Regardless of the type of model that we use, sample size is a big consideration. So larger sample sizes tend to yield more reliable results with fewer statistical errors, lower sampling variability, and higher out-of-sample replication. And some people have even said you need as many as thousands of individuals to capture reliable relationships. But it's not always feasible to have these large sample sizes, especially when we're considering clinical populations. So meta-matching is a transfer learning approach that was developed to translate phenotypic predictive models from big data to small data. So it relies on the assumption that in every big data set, there will be some phenotype that is likely to correlate with, even though it's not the same as a phenotype of interest in a small sample study. We can leverage that relationship to actually boost prediction in small sample studies. So the original paper was based off of UK Biobank data. They trained a model in a meta set of 26,000 participants and 33 phenotypes. And importantly, those phenotypes were all non-brain imaging phenotypes. And then they used those models to implement either basic meta-matching or advanced meta-matching using either fine-tuning or stacking to improve predictions. And the original model, all of these models are publicly available, by the way. The original one used just UK Biobank. I believe the most recent one actually also incorporated data from HCP and ABCD. So in this study, we used 3 independent data sets that were pretty much as different as we could get them. They were different in their collection sites, their demographic and diagnostic makeups, their measures of cognition, the imaging sequences that they used, and how those data were processed. And we wanted to see if we could use a meta-matching approach to characterize cognitive dysfunction in different clinical populations. Overall, we found that using this meta-matching approach, we are able to capture accurate and generalizable predictions of global cognitive functioning. So on the left, we have prediction performance, and in all cases, meta-matching yielded better results than kernel ridge regression models. And what was also interesting is that using this meta-matching approach, The models that were trained in one data set often could also generalize to other data sets. And not only were these models themselves generalizable, they were also largely robust, and we found that they weren't influenced by a specific diagnosis, sex, or age group. So here we have HCP on the left, TCP, which is the Transdiagnostic Connectome Project in the middle, and CNP, which is the Consortium for Neuropsychiatric Phenomics on the right. And at the top is just overall what those data look like in terms of the observed and predicted variables. And we saw that depending on the diagnosis, which is the second row, sex, the third one, or age, it doesn't actually influence it. So overall, they are very robust in what they capture. And finally, we looked at the specific relationships they were capturing, and we found that the brain networks that were implicated in cognitive functioning across all of these data sets were pretty similar overall. So we showed that increased connectivity within heteromodal networks and reduced connectivity between unimodal and heteromodal networks is predictive of better cognitive functioning. And given that this is consistent across these different data sets, which again, different in ages, sexes, diagnoses, it suggests that there may be a common transdiagnostic functional signature that might be indicative of cognitive dysfunctioning in different mental illnesses. Another factor we have to consider is who we're actually looking at in our studies. So our brains, behaviors, and relationships between them are highly influenced by a range of sociodemographic factors, such as sex, gender, socioeconomic status, age, environment, so on. In the context of psychiatric illnesses, this is important because, again, prevalence rates do vary across these groups. Here we have results for the sexes, so left is female, right is male. As you go up, that is age starting at birth, going all the way to 95 plus. And then we have the prevalent cases per 100,000 with different colors indicating different illnesses. So females overall are more likely to experience mood and anxiety disorders. Males are more likely to experience attention and conduct disorders. And these sex differences emerge pretty early in childhood and persist throughout the lifespan. And you might have also noticed that overall females do have a greater burden. So even though at some of those ages 5 to 9 and 10 to 14, it is higher in males. Throughout the rest of the lifespan, there is higher prevalence in females. It's not just sex. We also see differences across global regions and income levels. So on the left are different World Bank regions. On the right are income levels. Again, same diagnoses. And you'll see that the prevalence rates do vary based off both regions and income levels. So when we're thinking about these brain behavior relationships, it's also important to actually think about how these different factors might be contributing. And one way to do that is using these population-specific modeling approaches. So overall, when we're developing these models, we can develop it on everyone, or we can identify some aspect with which we want to split the data. So this could be sex, this could be diagnosis, this could be ethnicity, and that is a great way to actually identify relationships that might be specific to each subgroup and also see if those relationships are shared and if those predictions generalize across these different groups. So one example of how we've used this is we recently looked at how sex and gender shape functional brain networks in youth. And using sex-specific predictive modeling, we were able to show that gender, specifically parent-reported gender data in kids, can be reliably predicted based on functional connectivity data. So on the left, we have explained variance, right is prediction accuracy, females on top, males on the bottom, and each of those panels are showing self-report in blue and parent report in orange, and the asterisk denote that they were better than chance. And because we used a sex-specific modeling approach, we know that these relationships with gender are independent of sex because we removed that aspect of it. And then, like I said, we can also use this to gain a mechanistic understanding of how sex and gender actually influence the brain. So if we look at the associations between functional networks and sex, we see that there are connections within and between sensory motor and association networks that are involved. But then if we look at those associated with gender, they're very different. Gender, here it is female data on the left, males on the right. And again, warmer colors here are stronger associations, cooler are weaker associations. And I should mention that in this sex plot and these gender ones, the colors do not indicate directionality in any way. So it's not saying that one is more masculine or feminine. It's just saying it's associated with gender. So gender is predominantly related to functional connections within and between these different heteromodal association networks, including things like temporal parietal, default mode, limbic, and dorsal and ventral attention. We also see some slight differences between females and males. As you can see, temporal parietal has a stronger effect in females. Somatomotor has a stronger effect in males. So there are also differences in how the same behavioral phenotype might differently map onto the brains. I can also summarize these onto a network level. And again, you'll see that the sex and gender ones are very different. So using this population-specific approach, we're able to characterize how sex and gender map onto the brain and how those associations actually differ. And the same approach can pretty much be applied to study any other population subsample and any other behavioral phenotype of interest. So through these examples, overall, you can see that methodological choices do influence accuracy and interpretability, and there are specific strategies that you can use to overcome some of those challenges introduced by those methodological choices. So now I'll talk about a couple studies that we've done where we've leveraged these models to look at the neural underpinnings of specific aspects of psychopathology. And everything that I'll present is based on ABCD data, most of it with a sample size of around 5,000 to 6,000 individuals. So the first one that we did is looking at internalizing and externalizing behaviors, both of which are highly common in youth and associated with the future development of mental illnesses. Internalizing are those directed at oneself, externalizing are those directed at others, and both of these behaviors vary in terms of prevalence. Internalizing is more common in girls and women, externalizing are more common in boys and men. So we wanted to look at whether these behaviors map onto shared or distinct brain networks across the sexes. So again, we used a population-specific predictive modeling approach, sex-specific. We had 17 different behavioral outcomes we were predicting. The internalizing ones are in blue, externalizing in orange, series of other problems, as well as different scales that were DSM oriented, so more clinically defined. And we not only did we want to look at if we could actually predict these behaviors, we also wanted to see if our models would generalize both across behaviors and across sexes. So essentially, can you take a model that's trained to capture the functional networks that underlie internalizing behaviors in females and generalize it to predict, for example, attention problems in males? So all of this to say that there were over 1,100 different sets of models that we trained for all of this, and everything that I'll show you has been corrected for multiple comparisons across all of those. So looking at just the accuracy, so this is when the model is trained and tested within the same sex and behavior. We have results in males on the left, females on the right, and we're showing prediction accuracy, which is the correlation between the true and the observed and predicted variables. And again, the asterisk here denote predictions better than chance. So in males, all of the externalizing ones were predicted better than chance, as well as attention, social, and total problems, and similarly relevant scales within the DSM ones. In females, the results were largely the same, although there were a few that weren't able to be predicted. But what's really interesting is that there's also an internalizing behavior, specifically withdrawn depressed traits, that we were able to predict better than chance. So this is already telling us that there might be some sex differences in how functional connectivity is actually underlying these different behaviors. So in the next part, we, like I said, wanted to look at if those models generalize across sexes and across behaviors. So I'm not going to go into detail here, but left is models that were male trained, male tested. Right is models that were male trained, female tested. All of the vertically is the trained behaviors, horizontally is the test behaviors, and everything that's shown in color are those that survived multiple comparisons corrections. So what you'll see is that if you look at this part over here, it's basically telling us that if you're training models to predict one externalizing behavior, it'll generalize to predict another one. It'll also generalize to predict things like social problems, attention problems, and same thing for the DSM-oriented scales. what's really interesting is if you take those male trained models and test them in females, those models trained to capture externalizing or related behaviors can actually now predict an internalizing behavior. So essentially, this tells us that the same set of functional networks might actually be underlying externalizing behaviors in males and internalizing in females. We can check by basically doing the reverse. So here, female trained, female tested on the left, female trained, male tested on the right. Again, we see these patterns where the externalizing ones generalize across one another. But we also see that the model trained to predict withdrawn depressed traits in females generalized to predict these externalizing behaviors in males. And if we look at how they map onto the brain, positive associations are at the top, negative associations at the bottom, and on the left is the map for internalizing in females, and on the right is externalizing for males. So again, we can see that it is the same set of networks. So this suggests that the same set of networks that might be indicative of some aspect of psychopathology may actually manifest as different symptom profiles across the sexes. In other words, the presentation of different psychiatric illnesses might not actually represent fundamental differences in the neurobiology itself, but it might actually just indicate that males and females default to gender-specific, socially acceptable presentations of the underlying mental distress. So this has implications for how we diagnose and treat, because they're not necessarily different disorders, they're just showing up differently. So building on this work, we wanted to look at another early marker of psychopathology, which is psychotic-like experiences. So these are also highly prevalent in youth and represent early markers that can then indicate future development of either psychotic disorders, other mental illnesses, or in most cases, no disorder. And these are subclinical symptoms that include things like perceptual distortions, false beliefs, and disorganized thinking or speech. Typically, delusions are more common in girls and hallucinations in boys, but all of these experiences are pretty highly prevalent in both. In our data set, I believe we saw them in about 60% of all kids. And in terms of our predictions, we were able to use functional connectivity to predict both the total number of experiences that were reported by the kids, as well as the distress that they experienced as a result. So left is the total scores, right is distress, females in red, males in blue, and all of these predictions were significant. Again, we wanted to see if these experiences mapped onto the same or different brain networks across the sexes. So in the past study, we found that different behaviors, same brain networks. Here, what we actually found is same behavior, different brain networks. So across both sexes, we see that the limbic network is involved, but apart from that, in females, other relevant networks were temporal parietal, default mode, dorsal attention, and ventral attention. And in males, it's a bit different, where visual networks seem to be very important. And here, females are shown at the top, males on the bottom, and then we also have a little square for the non-cortical regions. So this tells us that these same experiences that boys and girls have might actually map onto different aspects of the brain. Importantly, the functional networks that we find to be involved with psychotic experiences differ from those associated with internalizing and externalizing behaviors. So this tells us that the relationships that we're observing that are influenced by sex also exhibit specificity in terms of the behaviors they're associated with. So we're not just capturing a general signature that indicates all psychopathology, it's actually specific to the symptom that we're looking at. And everything that I've shown you so far is using prediction models, but it hasn't been doing actual prediction because we're just looking at cross-sectional data. So now what we've done is actually use the functional connectivity data to predict future psychotic experiences in these kids. And those also work. So at the top is 1-year follow-up data, bottom is 2-year follow-up, left is females, right is males, red is total, and blue is distress scores in this case. And all of these models perform better than chance. In other words, you can take a child's functional connectivity data and actually predict whether a child will have these experiences up to 2 years in advance. As more data get released, we do want to see if this will also apply later on, and also if we can actually use that to predict a diagnosis rather than just these experiences. So overall, our results show that individual variations in functional connectivity may actually serve as a sex-specific prospective marker of psychiatric risk. So we might actually be able to use these to have early interventions. So everything that I've shown you so far has kind of looked at the prodromal stage, but there's a lot that actually happens in the premorbid stage that has huge impact on later functional outcomes. So the premorbid stage is when a lot of different factors associated with illnesses actually begin to shape risk in a longitudinal manner. So studying how different risk factors influence the brain during this stage can actually help us identify the mechanisms with which we can reduce that risk. So I'm not going to go through the details of the next, I think it's 5 studies, but we are doing a lot of work in this space, and I'm happy to answer any questions. I'm just going to highlight the main things we're finding. So in this first study led by my postdoc, Kathy, we're looking at how early life adversity across individual, familial, and environmental risk factors map onto brain structure. And what we find is that environmental risk factors, and specifically those related to social structural disadvantage, have a greater influence on brain structure than individual or familial risk factors. And this preprint should actually be up sometime today or tomorrow. So in the next study led by my postdoc, Lisa, we're looking at how recent stressful life events map onto brain structure. And here we're looking at the number of events, how they're perceived, so whether the children think they're positive or negative, and the overall impact that it has. And what we're seeing here is that it's not the number of stress or the amount of stress we experience, it's actually the amount of stress we experience that we perceive to be negative that is relevant. In this third study led by my third postdoc, Erin, we're looking at how age and puberty influence brain and cognitive development, and also if there are specific differences across the sexes. And what we find is that age and puberty have interdependent, but also independent influences on neuroanatomical and cognitive development. And these associations vary by sex, brain region, and cognitive domain, indicating a complex interaction between all of those. And finally, this is a preprint that came out maybe like a month ago. We looked at how impulsivity is related to neuroanatomy. So changes in impulsivity are a part of normative development. It does tend to increase in children, but it does also decrease after that. At their extremes, they can indicate psychiatric risk. So what we found is that impulsivity is linked to neuroanatomy, and these relationships vary across behavioral scales, so different measures of impulsivity, neuroanatomical features. Specifically, we looked at cortical thickness, surface area, gray matter volume, sexes, so between males and females, and time points. So here we considered baseline and 2-year follow-up data. And with that, this is a summary of everything I'll show you. And overall, I mentioned the stat earlier that 1 in 5 youth experience mental illness at any given time. By leveraging these large-scale data sets and machine learning models, we're hoping to better understand mental illnesses, how they emerge, who they affect, and how they might vary across different groups. We can also leverage these models to make individualized predictions. And the idea is that collectively, we can improve this 1 in 5 statistic, ultimately reducing individual and societal burden of mental illness. And with that, I'd like to thank my lab and my collaborators and my funding sources. Thank you.
Thank you, Elvisha, for a fantastic presentation.
Excellent presentation. I was interested in the text-specific functional connectivity results.
Mm-hmm.
So I noticed.
That's for the psychotic experiences, or
no, the previous ones.
The internalizing.
Yes.
Yes.
So I think it's very interesting. I was looking at the positive and negative associations, and I was wondering whether, because a lot of the regions that stand out here are part of this kind of theory of mind mentalizing networks. Could this, commonality, but distinctly associated with the externalizing versus internalizing factors, could it be related to the development of theory of mind processes?
Yeah, I think there's a lot like a lot of these patterns that we see with internalizing, externalizing psychotic experiences, all of these, they're very broad and dispersed. They're not just like a single region, which also indicates that they're not just affecting one aspect of behavior outside of them, they're having a broad impact overall. So and these regions are also ones that tend to develop later on during adolescence, similar to theory of mind. So depending on how internal and external influences shape that developmental process, that might be influencing both how theory of mind is developing as well as the expression of these behaviors.
So I was kind of like, well, how active is brain imaging ever going to be? Psychiatric elements. And so why don't you comment on, do you think brain imaging is useful? Like, do you think, and like, if not, or what would you combine it with so that you could actually have meaningful,
On its own, no. Combined with other data, yes.
like,
So you're gonna ask. This is a grant I just submitted last week, where our goal is to look at adversity in neuroimaging and mental health altogether, with the idea being that we can actually look at how adversity is shaping neuroimaging and adversity is shaping the brain, and those influences on the brain might be indicative of specific mental illnesses. So the idea is that adversity leads to brain, adversity and brain collectively lead to mental illnesses. And adversity is obviously just one aspect of risk factors. There's also many other ones that we could do. So theoretically, you could do this for everything else like genetics, other environmental factors, and have this more longitudinal causal relationship, which I think we will not be able to capture using just brain data itself. Yes.
Yeah, so
Sorry.
if you sort of necessarily expect activity that's sort of distilling down to some oscillations here, and then you say sort of how do you kind of understand these results from a sort of neurobiology cognition, being in visual, highlighting some automatic areas, some faces, and as you said, coming in. Can you kind of tell us what you think about from, say, a cognitive neuroscience or psychiatry point of view?
Yes. So do you want me to talk about the cognitive maps or these like more psychopathology related maps, or both?
Your interpretation, meaning of these maps in terms of the neurobiology.
So overall, what we're finding here is just how we can relate these behaviors to different aspects of the brain. And like I said, most of this has been done in a cross-sectional manner. And our idea is that if we can look at where these experiences are actually mapping onto the brain, we can then look at the downstream effects on things like cognition or also the emergence of different mental illnesses. So it's not necessary that the same maps will show up for cognition, but it does kind of give us an insight into how different experiences might be mapping onto the brain. The differences that we see across males and females here might actually also be related to the types of experiences that boys and girls have. So hallucinations are more common in males. We see a bigger involvement of the visual network in males. In this study, we did not separate it by the type of psychotic experience they were having. We looked at it broadly, but my NARSAT grant is actually specifically looking at if there is a symptom specificity to outcomes and also brain.
The sensory aspect of hallucination, and then some other sort of factors.
Yeah, so the limbic is one that we saw in both. It's hard to see, kind of like under there. So that might be underlying some of the more like emotional and higher order cognitive aspects of it. And we're hoping that as we have more data from future time points, if we look at specific categories of experiences, can we map those categories onto specific brain regions? So like, is that visual entirely driven by hallucinations, or is there also other experiences that map onto that?
Question then. So I'm wondering if at the one reason that I to be is that visual where their by state. So I was wondering if in the longitudinal studies that have you assessed the reliability of the measures that enter the predictive models?
So we're starting to look at that now as more of those longitudinal data are released. It gets very tricky because basically certain measures are self-report, other ones are parent report. There's also some that are teacher report. And then it's hard to know if it is a developmental progression in your behaviors or a reliability issue, like you said. So it's possible that a child that has a lot of these, I don't have the figure here, but we did look at if these psychotic experiences scores correlated strongly over time, and they were actually only like moderately to weakly correlated, but that's also what is expected in general because these experiences tend to be highly common in kids, but they get less common as you get older. So it's hard to know if it's a reliability problem or a developmental, like normative aspect. We're hoping that with this grant, so this is pretty much everything that we're going to be looking at. And for all of the adversity aspects and all of the mental health, we have multiple ways of those being assessed: self-report, parent report, teacher report. And we're hoping that we can actually do all these analyses using all of these different reported ones separately to see if those line up. Again, there's problems with that too, depending on what parents will report and what they won't, but it's the best we can do.
Also, I think functional connectivity based on resting state of MRI or based on task state of MRI also differs in its test-retest reliability.
Yes.
Also,
Yeah.
Good question. I wanted to hear more. Is that right on the models you used? Slide 70 to 73 on the external internal
Yes.
generalization. That is.
Doesn't think it. That's fine. Pretty much for most of these analyses, we are leveraging different ridge regression approaches. So I did at one point spend about 6 months trying to train a deep learning model to do the same things. It did not work. This was much better. Yes, we've tried it with functional, we've tried it with structural, as well as just like anatomical data.
Here's the pure model.
Yeah, so there's actually some cool work that's shown that, I mean, that's already starting with functional connectivity, but what they've shown is you can take functional connectivity data for an individual and then predict their task-based activity based on that data. And then using that task-based data, you can actually get better predictions for actual behaviors. So the idea is that using some baseline aspect of data from an individual, you might be able to make more comprehensive downstream predictions that you otherwise wouldn't be able to, which is primarily relevant in a clinical context where it might be hard to get patients in the scanner to do specific tasks. In terms of the rest of it, or given just how computationally expensive these models themselves are, so this is using the Schaefer 400 parcellation, so it's about 90,000 edges that are going into the model. It's already theoretically taking in a lot of data, but you're right that in that it could be taking millions of voxels more broadly. We're hoping that by summarizing it onto these functional networks and functional connectivity patterns, they might also be a bit more interpretable, because once you have the voxels, it gets tricky trying to figure out thresholds for clusters, what a specific cluster might mean if it's not broadly integrated to other things. So, that's fine. I don't have the figure here, but we did look at the trends in self-report and parent-report gender across all of them. Males exhibited more sex-congruent gender identities than females did. And the self-report is based on 4 questions that assess gender identity, gender expression, and gender dysphoria, whereas the parent report is based on 12 questions that assess gender expression and sex type behavior during play. All of these questions go from 0 to 5. So obviously the self-report had a much lower variability, especially because most of the kids were reporting scores that were pretty high across the board. We think that it might be due to variability in the parent report and that might be why we're able to capture these relationships. Our goal was to look at this at future time points, but that data is no longer available.
Any question? Any sort of like familial conflicts or sort of these environmental aspects that could be driving your patients that might be reflected in the difference?
Yeah, definitely. I mean, the parent report especially is obviously going to be highly biased by what a parent thinks is more masculine or feminine, which is where it gets very tricky. At later time points, they have to some extent assessed self-reported self-perception of data and self-reported others' perception, or self-reported self-perception of your gender and self-reported others' perception of your gender. So we could see a bit more signal there theoretically, but again, that data no longer exists.
Elvisha has a busy schedule today. Thanks again, Elvisha.